5 CMC mistakes that could delay or derail your IND ANDA approval. You have spent years building the science, the molecule works, the efficiency data looks promising, and then FDA puts your program on clinical hold. Not because the drug doesn't work, but because your CMC section had gaps. This happens more than most people want to admit, and in today's regulatory environment, strong science alone simply isn't enough to get across the finish line. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratory. Today, I'm going to walk you through the 5 most critical CMC mistakes that derail IND and ENDA submissions, and most importantly, how to avoid them. If you are a small or mid-sized pharma or biotech company navigating your first or second filing, this one's especially for you. Why CMC matters? Let's start with a reality check. CMC, Chemistry, Manufacturing and Controls, is essentially your promise to FDA that your drug is what you say it is, every single batch, every single time. Regulators aren't just reviewing your efficacy data, they are asking, can this sponsor actually manufacture this product consistently and safely? Quality data doesn't just check a box, it builds reviewer confidence, and when that confidence is missing, timelines slip, questions pile up, and sometimes entire programs stall. Think of it in this way: the path from your lab bench to market approval is paved with data. CMC is the foundation of that path. Crack that foundation early and everything built on top of it becomes unstable. Mistake number 1: unvalidated analytical methods. The first and probably the most common mistake we see is submitting with analytical methods that haven't been properly validated. Here is the thing: FDA doesn't just want to know whether you measured, they want to know that your measurement means something. Is it reliable? Is it reproducible? Does it hold up under different conditions, different operators, different days? ICH Q2R1 sets out very clear expectations for method validation: specificity, linearity, accuracy, precision, robustness. Yet sponsors routinely arrive at the IND stage with methods that were only developed for internal screening, never formally validated. The fix here is start validation earlier. Don't treat it as a submission formality, treat it as a scientific commitment. At Resolve Mass Laboratories, one of the things we do is integrate method validation into the development roadmap right from the phase 1, so things are no surprises when you are ready to file. Mistake number 2 is incomplete stability and impurity data. Now let's talk about stability and impurities, because this is where we see some of the most painful FDA queries. Your stability data is what supports your shelf-life claim. If that data is missing, incomplete, or based on conditions that don't reflect real-world storage, FDA will come back with questions. And accelerated studies alone won't cut it. You need long-term data too. Equally important, your impurity profile. Every degradant needs to be identified, quantified, and justified against ICH thresholds. We have worked on submissions where sponsors had excellent API data but hadn't adequately profiled their degradation products under stress conditions. That's the gap that's completely avoidable with proper planning. The takeaway here is stability and impurity programs aren't just regulatory requirements, they are your scientific argument for why the product is safe. Mistake number 3 is poor scale-up documentation. Here is something that surprises early stage teams. what works beautifully in a 2-liter reactor doesn't automatically translate to a 500-liter commercial batch. Scale up is where manufacturing reality hits theoretical process design, and FDA reviews batch reproducibility very carefully during NDA review. Every process change, every deviation needs to be documented with scientific rationale, not just flagged in a lab notebook somewhere, but formally captured with context, justification, and the data that supports it. Think of your scale-up documentation as the story of how your process matured. FDA reviewers are essentially reading that story. Make sure it's coherent. Mistake number 4 is weak control strategies. This one is a little more nuanced but critically more important. Control strategies that don't align with clinical risk. Under a quality by design framework, your specifications aren't just numbers pulled from a method. They are scientifically justified limits that directly connect to a patient's safety and product performance. When control gaps exist, when critical quality attributes aren't properly defined, or when acceptance criteria feel arbitrary, FDA will ask major questions. And major questions mean major delays. A strong control strategy demonstrates that you understand your product's variability and have designed your process to manage it proactively. That's the kind of regulatory confidence that moves reviews very forward. Mistake number 5 is poor data integrity and traceability. The 5th mistake might be the most underestimated. Data integrity failures, LCOA plus, attributable, legible, contemporaneous, original, accurate, and more, isn't just an acronym. It's the framework FDA uses to evaluate whether your data can be trusted or not. 21 CFR Part 11 compliance for electronic records, audit trails that are complete and unaltered, data that's traceable for raw instrument output all the way to your submission. These aren't optional extras. They are baseline expectations. Inconsistent records, overwritten data, or gaps in audit trails are serious inspection findings, and increasingly, FDA is scrutinizing data integrity not just during inspections but in the review process itself. How CMC experts reduce regulatory risk. So how do you navigate all of this, especially as a small or mid-sized company without a large in-house regulatory CMC team? The answer is partnership, and getting that partnership right early. Experienced CMC specialists don't just spot gaps after the fact, they identify issues before FDA reviewers do. They bring integrated planning that connects your analytical development, your manufacturing process, and your regulatory strategy into a single coherent narrative. At Resolve Mass Laboratories, we work with sponsors at all stages, from early IND enabling studies right through to NDA filing, helping build CMC packages that are technically rigorous and submission ready. The goal is always the same: fewer FDA questions, fewer timelines, and more confidence at every milestone. So let's talk about key takeaways from this session. Let's bring all together quickly. 1. Start CMC planning early. These aren't issues you want to discover at submission. Number 2, validate your methods before your major filing milestones. 3, build a comprehensive stability and impurity program from day 1. 4, document every process change and scale up decisions with scientific rationale. 5. Make sure your data is traceable, compliant, and inspection ready at all times. And if you don't have the internal capacity to do all of this well, partner with specialists who do. The cost of getting CMC right is a fraction of the cost of a clinical hold or a complete response later. If your team is working through an IND or ANDA submission and you are not confident about your CMC package, let's talk. Drop your biggest CMC challenge in the discovery, or talk to our specialists at Resolve Mass. And if you found this useful, please like and subscribe. We cover regulatory strategy, analytical development, and CMC deep dives regularly on this channel. And if you want to explore how Resolve Mass Laboratories can support your program directly, visit our website, connect with our specialist scientists, and we'll look forward to your discussion. Thank you so much for watching this session, and see you in the next one. Until then, goodbye.